Praveena from group 320. I am going to explain about the physics of protein biosynthesis. This one my topic is translation. In translation, the Crowell leaf model of tRNA explains how it structures, forms loops and helix, allowing a larger number of protein base pairing combinations. Like in this model, it separates spots leaves of the tRNA can shift independently, involving a number of possible arrangements. The concept of nature's game, highlighting the complexity of biological molecules like tRNA. And when the tRNA structure is unfolded by heat, thermal denaturation, it can return to its original form, a process called the renaturation. In study in 1972, Rich used X-ray analysis to study the structure of phenylalanine tRNA. He found that it has two helix segments connected at a right angle and forming a shape like the letter T. Translation is the process in which the genetic code from mRNA, which is copied from DNA, it used to create a proteins. Like the ribosomes are the machinery that performs translation. They are spherical particles that can only be seen under an electron microscope and are between the 15 to 25 nanometers in diameter. Ribosomes are made of ribosomal RNA. These rRNA 65% of the weight and proteins 35%. In ribosomes, in eukaryotes, complex cells like human cells are larger with an ATS sedimentation value. Eukaryotic ribosomes molecular weights about 4.1 to 4.7 million daltons. Like prokaryotic ribosomes are smaller with an 70s sedimentation value. The 70s ribosomes can split up to two smaller subunits like 50s subunit, 30s subunit. The layer 50s subunit has molecular weight of the about 1.8 million daltons, while the smaller 30s subunits weighs about 0.7 to 1.0 million daltons. In physical properties of E. coli ribosomes. E. coli ribosomes are about 20 nanometers in size and when dried, but they expand to about 30 nanometers. In this weighs about 3 million daltons and hold about 0.9 grams of the water per gram of ribosomes. If you talk about the ribosome structure, in ribosome structure, the 70s ribosomes from E. coli consists of two paired subunits, a larger 50s and smaller 30s subunit. In 50s subunits contains over 50 types of proteins and some rRNA. In 30s contains about 19 to 20 types of proteins and smaller RNA. In simple terms, E. coli ribosomes are made up of the rRNA and proteins. They can be broken down and reassembled in the lab. The proteins and rRNA have to come together in a very specific way for the ribosome to function. And for ribosomes to function in protein biosynthesis, the creation of proteins contains protein called transfer factors are needed. For the ribosomes to function properly in protein biosynthesis, it is necessary that special proteins be present in them, namely like transfer factors, which is bind to ribosome in the presence of a divalent cations, and these factors have been studied little. Divalent ions, the concentration is less than 0.005 m and are required for the interaction of the ribosomes with the mRNA or with the artificial polyribonucleotides. Each ribosome units only with one polynucleotide chain. That binding occurs in the 30th subunit. In the triple ribosomes, the mRNA like amino-acyl tRNA complex, the third component of the becomes bound by its anticode to the mRNA codon. The recognition and pairing also occurs in the subunit. Like translation begins in the initiation, that is the synthesis of the first peptide linkase. And if we talk about the initiation of translation, translation begins when the ribosome mRNA and a special tRNA called N-formyl methanoyl tRNA came together. And next, elongation, polycondensation. You can call it as a polycondensation. After the initiation, amino acids are added one by one to the growth polypeptide chain. And next, role of ribosome subunits. The TS subunit is responsible for binding mRNA and bringing the correct amino acyl tRNA. Next, ribosome opening and closing. As the ribosome works, it attracts between open and closed states. In the closed state, the new amino acid brought by the amino acyl tRNA in is positional close to the growing polypeptide chain, allowing the two to link together. And then protein synthesis, the formation of the peptide bond. In protein synthesis, the formation of peptide and when the ribosome is closed, a new amino brought up by the amino acyl rRNA is added to the growing protein chain. The carboxyl group of the existing protein chain is transferred to the amino group of the new amino acid. This forms a peptide bond. If you talk about the translocation, after the bond is made, translocation happens where the tRNA with the growing chain moves from the 30S subunit to the 50S subunit. This movement shifts to the mRNA along the ribosome. And next, the ribosome opening and closing. In this one, the ribosomes are open and closed like a mechanical devices. In polysome, it can have like 4 to 6 ribosomes for shift proteins like around 150 amino acids and 12 to 20 ribosomes for longer proteins. And mRNA degradation, the ribosomes are already attached, it will keep making proteins even it is a part of the mRNA start to degrade. And time and seed, it takes 30 seconds to make a protein with 400 amino acid, and the whole the protein biosynthesis is determined by the rate of transcription and translation, and these rates are commensurate since the mRNA labeled with radioactive atoms were observed a correlation between the lengths of labeled mRNA and polysomes, and there is no free labeled mRNAs present. In this one, overall, protein synthesis is a very efficient and coordinated process where many ribosomes works on the same mRNA